Welcome to the second installment of Fleener Teaches Biology via the Internet. This is yesterday's picture, which was showing a ligand being distributed across the body in order for the whole organism to communicate with multiple tissues at once. Of course, we refer to this ligand as a hormone, and this is a mechanism of long-distance communication. Today we're going to talk about another form of cell communication that's a lot more direct, and this is the communication involved in the immune system. Here we have a foreign agent, a bacterium, which we refer to as a pathogen, and it has a flag on it, and that flag is very specific, and our body is able to generate an antibody specific to that flag that recognizes it. I have nothing against Canada, I promise. I just happen to choose the Canadian flag. This antibody recognizes the flag, and it has our own body's flag on it that we can recognize, in order to destroy this pathogen. Now, this can happen in the human body. It can happen in all vertebrates. It can happen in any organism that has an adaptive immune response. And what this is all about is all about war. If you did not appreciate the fact that that was a TIE fighter from the movie Star Wars at the very beginning of the movie, I feel bad for you. The reason why I used that TIE fighter was because we're talking about war here. War between foreign pathogens trying to invade your body and immune cells trying to destroy those pathogens. And everyone has their own flags, and it's very important to recognize these flags in order to know who to destroy. So the flags themselves are called antigens. A specific bacterium from a specific bacterial species, or a specific virus, or a specific fungus, or a specific anything that shouldn't be in your body, including specific human blood cells that are of the wrong blood type, have antigens on them. And we raise antibodies to recognize those antigens so we can know what to destroy. So specifically, an antigen is like a flag on different types of pathogens. And we have cells called B cells, which are part of the humoral response. Our B cells make antibodies that recognize the antigens. So these antibodies have a very specific shape, and they very specifically recognize the shape of the antigens present on foreign pathogens. And they too have their own little flags on them that our immune cells can now recognize. Once the antibody recognizes the specific flag, then we have the cell-mediated response, in which cells called T cells, which are white blood cells, they go through your bloodstream, they see anything with this antibody attached to it, and they kill it. But it doesn't actually look like a cat with a machine gun, as cool as that would be. It actually, there you go, it kills these things. These guys have yet to be killed because they don't have the antibody on them. Doesn't actually look like a cat with a machine gun, it looks like a cell. A T cell is a cell, a white blood cell. And it has an antibody receptor on it. So this receptor can recognize anything with the antibody flag on it. And remember, the antibody itself recognizes the flag of a foreign pathogen. So it's a huge system of cell communication. Here, this antigen on this foreign pathogen, it itself is a ligand. The antibody here is a receptor that binds to this ligand. But the receptor itself, the antibody itself, has its own ligand side of it. The antibody is both a receptor side and a ligand side. And that ligand can be recognized by an antibody receptor on the presence of T cells. And that will activate a secondary messenger upon binding, which activates genes in the nucleus. Specifically, it activates poison genes, which will then be released in the form of poison proteins that will kill the foreign pathogen. So you have two systems here. You have a humoral system, which has B cells that make antibodies against the antigens. They make this antibody with the flag on it here, recognizing specific antigens. And then you have T cells, which travel throughout the bloodstream, and they recognize antibodies these antibodies, and they kill any pathogens that are attached to them. Just like that. So, my question is, explain how cells of the immune system communicate with other cells. Not how they, no, just how they communicate with other cells. This is the key word here. How do they talk? How do T cells, for example, talk to pathogens? It's not a very friendly conversation, I'll tell you that much. Okay, so the antibodies that are made are custom made for every antigen. So you can imagine there's millions upon millions of different bacterial or viral or fungal species that may or may not cause harm to your body. And every one of them has different antigens on them. So every time a new agent comes into your body, you have to make a new antibody that's specific to it. And the ability of your body to make antibodies is something I've never understood. It's something that's taught. I just never took the class. But it's really fascinated me. And I've remained fascinated in ignorance for a long time now. But here's just a few examples of different antibodies that can be made. And basically any possible protein, which is what an antigen usually is, you can make an antibody that specifically recognizes it. Just like an enzyme specifically recognizes a substrate, you can make an antibody that specifically recognizes an antigen, or a receptor specifically recognizes a ligand. You can make an antibody that specifically recognizes an antigen. In fact, in the lab, if you're interested in a protein, like I was interested in a very specific protein, one thing you could do, which requires a fair bit of money, but you can still do it, is you can get another lab to make you an antibody specific to that protein. And the way they do it is they take the pure protein, they inject it into rabbits or goats or sometimes donkeys or sometimes mice, and they have the immune system of those animals generate antibodies specific to that foreign protein that they've injected in. And then it's a very horrible process of actually causing the animal to bleed out, extracting blood until the animal is completely dry and dead, and generate all the blood it can, and then you harvest the antibody from that blood. But then you can use that antibody to extract your own protein in experiments, and it's quite cool doing that. So actually using the antibody in the lab is wonderful. Generating the antibody in a lab is not so animal friendly, and I'm really glad I never had to do that. So once you have the antibodies in your body, you're never going to get rid of them. You're always going to have them. And that's what's really cool about the immune system. And you guys probably know this already, that, you know, what doesn't kill you makes you stronger. And the whole explanation behind that is what doesn't kill you allows you to generate antibodies against it. So if a foreign pathogen comes into your body, and a pathogen is anything that can cause disease, so any foreign bacterium or virus or fungus comes into your body, or protozoa, or I guess anything. I'm not really sure what can cause disease, or maybe even a multicellular worm. Good gosh, that's no, your immune system wouldn't cover that. Anyway, so anything that comes into your body, you can make an antibody against it, and those antibodies are forever in the memory of B cells. And once you've made antibodies from a B cell, you're always going to be able to make those very, very quickly. So you've never been exposed to a pathogen before. You get exposed, B cells make a bunch of antibodies, and they have to kind of do some tinkering in order to make those antibodies. Then you end up killing the pathogen, and you survive. Good for you. And then here's the second time around. Maybe you have a really big fever, and that kills all the or that denatures all the enzymes of that pathogen, and the pathogen dies, and you survive. Good for you. Then the second time around, you get exposed to the pathogen. There's already some antibody present in your body, and the B cells, because they have this memory, part of the humoral response, they're able to make a bunch of antibodies super, super quick. In a short amount of time, they can make really quick antibodies, and then your white blood cells are going to be boom all throughout the immune system, ready to kill and eat anything. and everything's going to be great. The exception with this is HIV. That's what makes HIV so bad, is because it specifically infects immune cells and inhibits immune response. So people with AIDS often die not because of AIDS, they die because of something like the common cold, because their immune system is so compromised. And that's how we're able to make vaccines. Vaccines are just dead pathogens. And you can imagine if you I don't know, heated up some pathogens, killed them, they would still have the antigens present on the outside of their body. You know, a dead body is still a body. And so you can inject those in, to preferably younger the better, inject them into a person or an animal, and the person's B cells are going to recognize those antigens. Even though those antigens are attached to dead bodies, the B cells are going to find them as foreign antigens and develop antibodies against them. And that way, when the real live thing comes in, like the measles virus, I think it's, yeah, surely it's a virus, comes into your body the next time, ready to kill you, you're ready with all of your memory of your B cells and the antibodies there to make a bunch of more antibodies and have your T cells, part of the cell-mediated response, go and kill them all. Vaccines are one of the most important things to ever happen to modern medicine, because they effectively prevent diseases by making our immune systems prepared for these really, really terrible diseases that would otherwise kill us. And blood types are also an example of the immune system, because blood types themselves are antigens. People who are type A have a type A antigen on them. People who are type AB have a type A and a type B antigen present on the surface of their blood cells. People who are type B have only a type B antigen present on the surface of their blood cells. And people who are type O have no antigen present on their blood cells. So think about this. If you are a type A person, for example, and type B blood is given to you in a blood transfusion, your B cells are going to recognize that type B blood as a foreign agent, potentially a pathogen. It doesn't matter that it's human blood. It doesn't matter that it's eukaryotic blood. Your B cells will recognize it as a foreign agent. It will generate antibodies against the type B blood, and the T cells from the cell-mediated response will go find them and kill them. So that's why if you get a blood transfusion, it has to be a very specific type of blood. But if somebody gives you type O blood, no matter what you are, type O means there are no antigens present on those blood cells. So your B cells will not make any antibodies against them, and they will be able to be used to transport oxygen and nutrients throughout your bloodstream. So that's why type O is the universal donor, because it does not have any antigens on the present on the surface of the blood cells. And that's the same concept with organ transplants and organ donations. Organ transplants often fail because of the immune response. Basically, you put this huge organ in, even though they're human cells, human heart cells, for example, or human liver cells, and the immune system recognizes those cells as foreign agents. They generate antibodies against them, and they kill them. They kill those cells, and that's, you might hear like on Grey's Anatomy or something. It says their immune system is rejecting the transplant, and that's because the immune cells are killing the transplant, and that's why you have to have a very specific match. Doesn't matter that you're willing to give your kidney. You have to be a specific match, meaning you have to have the right antigens that body is going to not want to generate antibodies against and kill in order to give that transplant. And when organ transplants are done, doctors treat their patients with drugs called immunosuppressants, and that is exactly what you'd think it is. These are drugs that suppress the immune system, so it inhibits the ability of T cells or B cells to generate antibodies and T cells to recognize and kill those pathogens attached to those antibodies, and the organ eventually gets accepted, I guess. I'm not really sure about the medicine behind that, how the organ eventually gets accepted. But yeah, anyway, okay. So here's what I want you to do. Create anything you want, any kind of representation, showing how the vaccine prepares the body for a fast immune response to a specific pathogen. Remember, a vaccine or the exposure to any kind of pathogen gives a faster immune response the second time. So I want you to show some kind of representation that shows why that is, and how the vaccine is able to do that. And for the closure, compare the immune response to cell signaling. So we talked about cell signaling on Monday and Tuesday. Which components are analogous? That means an analogy. So what part of the immune system is a ligand? What part of the immune system is a receptor? And what part of the immune system is a cellular response in response to the activated receptor? Think about that and email me or remind me any questions. Thank you very much. Goodbye. Oh yeah, goodbye. Star Wars.